So welcome to our first Bioscience LITE event. So this is a collaboration between the public engagement team here at the Wellcome Genome Campus and the public engagement team at the Bioscience Institute as well. So I'm just going to give you a very quick introduction to the campus and the programme, and then I'll hand over to Tristan, who will be here shortly, who's going to give you an insight into how he uses stem cells to model human disease. And then we're going to go of a tour of our cellular and phenotyping department, which is really exciting. It's a really, really large-scale building. That's Dr. Celine Gomez is going to be doing that for us. And then towards the end of the evening, we're going to have a little discussion on what are good or not so good stem cell resources, anything that you would recommend to colleagues. Yes, so hopefully if you've got any favourite stem cell teaching resources, please do let us know and we can discuss that in groups. So a little bit about where we are. This is the Wellcome Genome Campus. We've been on this campus since 1993. This is the UK home of the Human Genome Project. The public engagement team is responsible for all the public engagement on this campus, so that means we represent both the Sanger Institute but also the European Bioinformatics Institute as well. So we have over 1,200 people working on this campus from all sorts of backgrounds, covering all sorts of STEM subjects. So the main focus of this campus is to use genomics to understand human disease and health and also pathogen variation and to manage big data as well. So that's a key thrust of what we do, but we have a whole facility just dedicated to stem cells as well, and that is a fundamental part of what we do too. As a sort of what can actually both campuses, not just the Wellcome Genome Campus, but also the Bioscience Institute, what can we offer you as teachers? Well, we both offer sites visits, so you can bring your schools onto our sites and find out from our scientists about the work that happens here. We also have a range of online resources that are available for teaching, which can support your teaching in the classroom. Babram currently have a fabulous STEM Insight programme where you can actually spend a week there going into the different labs and finding out more about the research. Hopefully this is something we will be introducing maybe next year. You have a leaflet in your goodie bags about this if you want to find out more. We also at the Wellcome Genome Campus also offer CPD on genomics as well. So if you want to find out a little bit more, if there's little areas that you're not sure about, we can offer specific training on that topic as well. So if you want to bring your colleagues along, we can do bespoke training for you. So a key resource, if you want to find out more about genomics, but also stem cells, we have a new page on that, is this website here. Are you familiar with this website, some of you? Great. So it's yourgenome.org, and it's very, very useful resource. Now, in your goodie bags, you have some postcards like this. And these, on the back of these, are some examples of some of the key resources, fact pages, story pages on your genome that are relevant to a range of different curriculum-specific areas. So we have DNA sequencing, infectious disease, cancer, human disease, DNA, genes and genomes, and working with animals. And these are going to be some of the themes that are going to be explored in the Bioscience LITE programme as well. But your genome has a range of classroom activities as well, ranging from things from primary schools all the way up to post-16, which you can download for free. It's all completely available. But we also have a really lovely range of 3D videos, animations, videos, covering a range of topics from DNA sequencing, DNA replication, DNA to protein as well. And again, these are all freely available. You just need to download them as well. So hopefully these will really help support your teaching in the classroom as well. I've got great pleasure now introducing you to our first speaker, which is Tristan, who is here. So Tristan works in a very interesting area, working with induced pluripotent stem cells. So I'm not going to sort of tell you too much more because that'll be stealing his thunder.
Well, good afternoon, everybody. It's my absolute pleasure to be here today to talk to you about how we use stem cells to model human disease. Some of the material I'll be covering today might be a bit technical. I'm more than happy to answer questions mid-presentation, so please feel free to shout out. I'll start with a bit about myself. So I received my BSc in biology from UCL, and in 2013 I was awarded my PhD in cell molecular biology from Imperial College London. During my doctoral training, I focused on the interactions between mammalian cells and invasive bacteria, and this was at the cellular level. And it's here that I developed a strong interest in the cellular biology. This interest led me to a Cambridge-based startup called Bioscience in 2014, and this is where I first became introduced to the awesome power of stem cell technology. After a very exciting spell that lasted just under 2 years, I joined the Sanger Institute in January 2016, where I'm currently head of the high-throughput gene editing core facility. So who are the high-throughput gene editing group, and what exactly do we do? Well, in short, this group is made up of a collection of scientists across many different disciplines, including genetics, cell biology, and computational biology. And as a core facility, our role is to support the research interests of the Institute whilst adhering to Sanger's vision of science at scale. More specifically, we are generating gene knockouts at scale in a type of pluripotent human stem cell called the induced pluripotent stem cell. And to create these knockouts, we're using a revolutionary gene editing technique called CRISPR-Cas9, which I'll be returning to a bit later on in this talk. To date, we have assessed over 454 genes and have been successful in creating around 150 knockouts. So you've heard me mention stem cells now on a couple of occasions, but what exactly are they? Have most people in the room heard of stem cells before? Yep. So stem cells can divide over and over again to produce new cells, and the appeal is that when they divide, they can change into other types of cells that make up the body, and this includes neurons, cardiac cells, and liver cells, for example. And these stem cells can be divided into 3 main types. You have your embryonic stem cells, which most of you might be familiar with. There are also the adult stem cells, which are very similar to embryonic stem cells, except that they're limited by their differentiation potential. And finally, there are the induced pluripotent stem cells, or IPS cells, which are a revolutionary type of stem cell and the cell type that I'll be focusing on for the remainder of this talk. So what are IPS cells, and why is there so much interest in them as a research tool? Well, unlike embryonic stem cells, which are generated from embryos shortly after fertilization, IPS cells can be generated from any adult cell, including skin or blood cells. Just like ES cells, they can change into various cells in the body, they can proliferate indefinitely, and excitingly, from the field of cell therapy, there is no immune rejection. So if you were to take skin from a person, turn that skin into IPS cells, and put those IPS cells back into the same person, you won't have any incidence of immune rejection. With no standardized way of making and culturing these cells, there is a concern around quality. However, with this technology, we have been able to overcome the ethical concerns that I'm sure you're familiar with embryonic stem cells. So, scientists are taking adult cells like skin and blood, and through a process called reprogramming, are turning them to become stem cells. And they do this through the short-term introduction of a small number of transcription factors. Now, these transcription factors are proteins, and they function normally to ensure that the right genes are expressed in the right cells at the right time. Collectively, these transcription factors are called Yamanaka factors, and these are named after the researcher who identified them, Shinya Yamanaka, who received the Nobel Prize for his work on IPS cell technology. And what you see during this process, if you're lucky, is the emergence of IPS colonies shown there on the right, which we can pick, we can isolate, and we can expand indefinitely. And because these cells are pluripotent, we can differentiate them into specialized cell types, such as heart cells, liver cells, neural cells, pancreatic cells, etc. And we're fortunate here at the Sanger to have so much expertise in a technology that is only just approaching 10 years old. The Sanger has been contributing towards the generation of a UK national IPS cell resource for a number of years now. And this project is called HippsKey, or Human Pluripotent Stem Cell Initiative. And this was established in 2012 to address the community requirement for a large, well-characterized collection of IPS cells for use in research. And the project's overall strategy is to derive 2 or 3 candidate IPS cell lines from each donor, healthy or diseased, and to generate an initial set of genetic and phenotyping assays on all candidate lines. This project has taken around 4 years to complete and has required a multidisciplinary approach with many different collaborators who are specialized in different areas, such as the generation of cell lines through to data analysis. But why do this? Well, if you have a condition such as cardiovascular disease, which can have over 100 mutations that could contribute to its cause, how do you measure each gene's role? Animal models are unlikely to help you with this because their changes may be too subtle, and individual IPS cell lines are genetically and phenotypically heterogeneous, i.e. they're different. So if you're studying stem cell lines from one individual, that wouldn't tell you the whole story, as people carry different genetic risk factors. And so the solution has been to generate IPS cell lines from hundreds of individuals and to assess the physiological variation. So what they did here at the Sanger was to set up an experimental pipeline to systematically generate IPS cells. And the focus here has been on the characterizing the phenotypes related to natural genetic variation. And the output has been a very comprehensive, high-quality reference set of IPS cells from healthy volunteers. There are around 711 IPS cell lines that have been generated, and these have been from around 301 healthy donors. These cell lines are also readily available from 2 cell banks based here in the UK, ECAC and EBISC, for use by the wider research community. I just want to draw your attention to this publication that has been recently published for the HippsKey project, the key finding of which is that 46% of differences observed were due to differences between individuals. And what this data will allow researchers to do is to put disease variation in context with healthy individuals. So one of the most exciting applications of IPS cells is their use in disease modeling. Many disease-relevant cells, such as neurons, are not easily accessible in patients, and therefore animal models, especially mouse models, have been widely used to understand the pathogenesis of human diseases. However, mouse models do not always recapitulate the phenotypes manifested in humans. In this case, patient-derived cells can be reprogrammed into IPS cells, and these IPS cells will be carrying a disease-specific genetic mutation. These cells can then be differentiated into the disease-affected cell, such as neurons if you're interested in neurodegenerative disease. And importantly, these disease models can be used to identify compounds that alleviate disease pathology in vitro. And very excitingly, especially for the Sanger, is that gene technology is now in a position that enables us to study and model diseases with very high efficacy. And one such technology in particular, called CRISPR, is regarded as a game-changing genetic engineering technique. Indeed, since 2011, the number of peer-reviewed papers with CRISPR in a title or abstract has increased by 1,453%, with no indications of slowing down. So what exactly is CRISPR? Well, CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. And this name essentially refers to the way that short, repeated DNA sequences appear in the genomes of bacteria and other microorganisms. CRISPR sequences are a crucial component of the immune system of these simple life forms. Just like us, bacterial cells can be invaded by viruses, and if a viral infection threatens to, if a virus threatens to infect a bacterial cell, the CRISPR immune system can thwart the attack by destroying the genome of the invading virus. So how does it work? Well, CRISPR regions are composed of short DNA repeats, shown here by the black diamonds, and spacers, shown here by the colored boxes. When a previously unseen virus infects a bacterium, a new spacer is incorporated amongst the existing spacers, and this CRISPR sequence is then transcribed and processed to generate short CRISPR RNA molecules. The CRISPR RNA guides bacterial molecular machinery to a matching targeting sequence in the invading virus, and this molecular machinery is responsible for cutting up and destroying the invading viral genome. And we refer to this molecular machinery as Cas9, which is a protein that essentially behaves as the molecular scissors for cutting DNA. So you might think at this point, well, we've been here before with gene editing, so what's all the fuss about this approach? Well, for one, it's very cheap and accessible. It has unprecedented precision, efficiency, and flexibility. Because CRISPR has a strand of RNA that guides the target DNA into the enzymes, the system effectively functions as a GPS, while other genetic engineering methods work off essentially a rough map. We can use this technology in any organism, and it overcomes the taxing genetic breeding strategies. Now, I said it was cheap and very accessible. This is actually on Amazon. So this is a DIY bacterial genome engineering CRISPR kit. So it just shows you how easy and cheap it is. So how are we using it? Well, the most important aspect of the CRISPR-Cas9 system is the introduction of a targeted double-stranded break with high efficiency. Because non-homologous end joining is the prominent double-stranded break repair pathway, and because this repair pathway is error-prone, you get these insertion or deletions, which can lead to the introduction of small random mutations. Double-stranded breaks can also be repaired by a process called homology-directed repair, and this is a process where homologous recombination, where a DNA template is used to provide the homology necessary for precise repair. The efficiency of this process, however, is substantially lower in comparison to non-homologous end joining. So to generate a knockout, we first force CRISPR-Cas9 complex into IPS cells. We then take these cells and plate them at low density. Each cell will be modified slightly differently and so represent their own unique populations. It is common for some cells to mix with each other, forming what we call mixed populations, and this is what we look out for downstream to exclude them from analysis. These single cells then form colonies, which are visible by eye and that we can pick and isolate for further expansion. And through a process called genotyping, we can then determine the genetic makeup of these cells at the specific gene that we targeted. And overall, we aim to obtain a knockout that is homozygous, but we also see heterozygous knockouts and those that haven't been targeted, which we called wild-type cells. So I know that's a lot to take in regarding CRISPR. So just to recap, CRISPR technology relies on two components, an enzyme and a guide molecule. The guide molecule takes its enzyme to a gene that you want to modify. The enzyme cuts the gene, and then it can be repaired in many different ways. You can either change the function of the gene, you can take it away completely, or you can even make the gene more active. And what we're doing here is working to disrupt the function of the gene. Currently, all of our knockouts are generated in one healthy background, and this is called the COLF2C1 IPS cell line. And this cell line was obtained as part of the HippsKey project and has been extensively characterized. We have whole genome sequencing for it. By stable carrier type, we mean that genetically it's stable over many passages in a 2D culture environment. And there's also open access rights to it as well, so we are able to share it with our collaborators. And the benefits of using one workhorse IPS line is that it allows researchers to create what we call isogenic models. And this is where you have a mutated cell line and the accompanying normal cells as well. In addition, because we are only using one cell line, researchers don't need to worry about the genetic variability. And so it allows them to essentially determine the causative relationships between genotype and phenotype. Finally, our cell line has a broad differentiation potential, making it appropriate for disease modeling. And we are fortunate here at the Sanger to have many groups with expertise in differentiating IPS cells, and we've been encouraged by some of the data that has been coming through. And I'll be sharing that with you now. So the first example I have for you is macrophage differentiation. So macrophages play a significant part in the immune responses. They assume a defensive role exhibited by their ability to carry on phagocytosis. So here at the Sanger, the pathogens group are using our IPS cell lines harboring genetic mutations to study how the functions of genes during the interactions with pathogens. And these pathogens could be salmonella or chlamydia, for instance. Now, salmonella and chlamydia are very efficient at infecting host cells. So what interests the pathogens group here is the question of resistance, i.e. what is it that makes macrophages resistant to being infected by these very efficient bacterial pathogens? And so what we've done is provided them with knockout lines with the genes that they think are responsible for this process. They're turning these cells into macrophages and then looking at whether or not their susceptibility to infection increases or decreases. Next are intestinal organoids. So organoids in general are very hot at the moment. These organoid cultures are three-dimensional. in vitro tissue models that incorporate many of the physiologically relevant features of the in vivo intestinal tissue. And this near physiological 3D model facilitates an accurate study on a range of biological processes, including tissue renewal, tissue responses to drugs, and also mutation and damage. And again, the pathogens group are using these cells to look at host-pathogen interaction. And we've actually got an EM photo that they've taken here. So this is looking at Salmonella typhimurium, and these are the intestinal epithelial cells. So this is quite a neat photo of showing basically the beginning of the invasion process. And so what they're doing in this case is taking normal and mutant IPS cells, creating intestinal organoids, and looking at how these genes play a role in facilitating infection by the bacteria. And yeah, in this case, they're looking at Salmonella. The final example I have for you is from my group, where we've been able to set up a protocol to generate neurons as part of our collaboration with the DDD study. So the DDD study is a partnership that is run here at the Sanger with the aim to advance clinical genetic practice for children with developmental disorders. You may have seen the recent output on the BBC website, where the study had actually identified 14 new developmental disorders, all caused by spontaneous new mutations not found in the DNA of either parent. And so one aspect of the study is the generation of mouse models to look at the developmental disorders. And to complement these mouse models, we've been asked to generate knockouts of the same genes but in IPS cells. And these cells will then be differentiated into the relevant cell type, in this case neurons, to ask the question of whether they have any therapeutic potential. So overall, IPS technology has created powerful new opportunities for modeling human diseases and offers hope for personalized regenerative cell therapies. However, as you can see here, there are a number of challenges that still remain. For instance, there are a lack of standardized protocols for specific applications. We know that there are excessive apoptotic and spontaneous differentiation signals that are secreted during the cell processing stages. So this can lead to spontaneous cell death, but it can also lead to the differentiation of the IPS cells to an uncontrolled cell line, terminal cell type, for instance. As I mentioned earlier, there's questions around quality, so the impurity or heterogeneity of the propagated cells. There's a question around how genetically stable these cells are long term, and also there is the question of their tumor geneticity when it comes to cell therapy applications. So it's important to remember, however, that the growth of any type of mammalian cell in vitro requires growth media, extracellular matrices, and also environmental factors, and all of these can have an influence on the above issues. And so maintaining high-quality IPS cells is absolutely essential because subsequent applications would depend on your starting material. And what I've shown here on the right is just a schematic of all the optimizations that we've been going through over the last 17 years or so, trying to get to a point where we can standardize IPS cell culture. And at this point, I'd just like to pass over to Fran, who's going to help discuss how the relevance of the work that we do to the curriculum.
So Tristan and I had a chat, and we sort of looked at some of the curriculum links that were out there, and how some of the topics that he's sort of just mentioned can really relate to what you're teaching in the classroom, because he blends a little bit of genomics and stem cells. It can touch on quite a few aspects of not just the GCSE curriculum, but a lot of some of the new A-level specs, so obviously the IPS elements of it. You also do a little bit on Alzheimer's as well, don't you? Which is obviously part of that. But particularly the fact that, you know, there's the genomics and the IPS cell lines blending together, and the use of biotechnology. All of this is quite curriculum specific. So if any of you have any questions around that, or you want more resources on that, let us know, because that could be areas where we could be sort of looking into finding more things. But also at the A-level spec, you know, the fact that students have to do EPQs, this is quite a broad area, and there's a lot of really gritty, interesting questions that could be asked and perhaps, you know, answered by sort of looking deeper into this. So this is potentially a really exciting EPQ area. So again, this could be something that we want to discuss a little bit later on, potentially. So these are just some of the things that we sort of picked out we thought were quite interesting. You may have your own ideas about how it links into the curriculum. Are you doing an EPQ project with a student at the moment?
Yes, I am. So they're actually looking at the CRISPR technology, and I showed before how obviously you can get these kits available on Amazon. Quite frankly, it seems quite scary now. The fact that we don't really know the true potential of this technology, and it's really kind of set off in the last few years. And so she's actually looking at the question of kind of the ethical side of using the CRISPR technology, and I've been assisting her in terms of developing the question, the material to look at, providing publications, etc. to look at as well. And with that, I'd just like to say thank you for your attention, and to say thanks to everyone that's helped to do the work.
Are there any questions?
Okay, you use CRISPR to guide the RNA.
Yeah.
Sorry, to guide the enzyme to cut.
Yeah.
So when you're creating a knockout, you cut it. And then you obviously have to mend it again.
Yes.
But so, do you cut a bit out?
So what we're actually doing is relying on the host's own ability to repair the cut.
Yeah.
So there are, in your own cells, there are naturally occurring DNA repair pathways. All we're doing is introducing the cut, and then we're relying on the repair pathway to introduce the mutation. So the most frequent repair pathway is non-homologous end joining.
Yeah.
And that can lead to the insertion or deletion of some bases.
Okay.
And depending on whether that's in frame or out of frame or where it falls, it can disrupt gene function.
So it doesn't always create a mutation?
Doesn't always.
Okay,
well, it depends really on where you're targeting.
Right.
So there are a number of factors. The protein, how pure it is that you're using, how active it is, the RNA, how stable it is, and also the gene that you're looking at, whether it's easily accessible or not.
Yeah.
It's quite a technically challenging process, but if you are able to target the locus, then you should be able to get.
And then how do you know that you've produced a knockout?
So at that point, what we're doing is basically sequencing a very small area that we have targeted to look at what. Basically, what you're looking for is whether it's been targeted on both alleles of the DNA or whether it's just on one. And we use next-gen sequencing for that.
Right. Okay.
So we're actually quite fortunate here that we've got the DNA pipelines facility. So what they're doing is, or what we're doing initially is PCR amplifying our small region that we've edited, and then they're basically sequencing it, and then we kind of decode it and have a look at it. Yeah.
Okay, lovely. And then just because can I just I presume the Human Genome Project has informed this a lot in terms of being able to look at the genes you want to knock out.
Exactly. Yeah. We can only go by a reference, so what we're doing ourselves is actually designing these CRISPR RNAs, and so we have to go to the human reference in order to actually get these designs. Human Genome Project was based off of one human reference. We're now finding that there's obviously differences between each individual, which is why it's now gone to the 100,000 genome project. So we do find that some CRISPRs don't work. Now, whether it is because the reference that we are using isn't quite correct in terms of what we think it is possible.
Gosh, amazing. Thank you.
Anybody else have a question? So you've been generating, obviously, neuron cells. How does that sort of have an effect on understanding diseases like Alzheimer's? Is there some work that you're doing on that? Yeah,
so there's actually, within the gene editing department here, there are two groups. The other group are actually focusing on a very small mutation, so a point mutation, which is basically taking one base and changing it. And you'll find that with a lot of these neurodegenerative disorders, it is just a single base change that is leading to their onset. So what we're doing is creating knockouts in the genes that we know are associated with these diseases, and this kind of gives us a high-level functional output. And then the other group are actually looking at more of the actual relevant cell type, so introducing a point mutation, and again looking at whether or not you get an output similar to what you'd expect for Parkinson's or Alzheimer's. And so basically what we're doing at the moment is creating the IPS cells, and then because we've now optimized this neurodifferentiation protocol, we can then take these cells to neurons, and then just in a cell culture dish can start to reproduce what we expect the behaviors to be for Alzheimer's or Parkinson's, for instance.
Could that lead to potential new drugs, or help understand how these cells respond to drugs?
Yep. I mean, so obviously it's very difficult to get patient neurons. So one of the things that you can do, obviously now with this IPS cell technology, is you can take a person who is known to have Parkinson's disease, you can take their skin, you can convert those into IPS cells, and then those IPS cells can be turned into neurons, and actually those neurons will behave very similarly to the neurons in the person's brain. And you can start to get an understanding of what you should be looking for. With this gene editing approach, we can kind of bypass that, because at the end of the day, when you're taking a person's skin and you're essentially using an IPS cell line or a donor line that you're not very familiar with, whereas with our COLF line, we've whole genome sequenced it, we're quite confident in the sequence of it, and we've well characterized. So we're quite confident when we create the mutation that we'll have a good system that can run in parallel.
So you're talking about a point mutation causing Alzheimer's. That's a bit scary, actually, isn't it? But presumably that means that would have happened really early on in the embryo. If that mutation, it's not going to happen in the brain when you're older, because otherwise every cell would have to mutate the same way, wouldn't it?
Yeah. Well, I mean, obviously there's the genetic factors to it, but there's also research out there now showing the environmental impacts as well. So for instance, if you're a footballer and you're heading the ball 10 times a day, there is the research to show that can lead to neurological issues as well. Okay. So I think we're using these IPS models not only to look at therapies, but we're also using them to look at just to improve our understanding, if you like.
Yeah.
I mean, you talked about how we can potentially come up with cures. I think one of the difficult aspects of using IPS cells is that they're fetal-like. So when you get the neurons, they are fetally behaving. And you have to culture them for a very long time, sometimes up to 300 days, to start getting the mature functions that you'd see from primary cells. So that's one of the technically challenging aspects of using them, and that's why these big pharmaceutical companies have struggled to really bring them in as a model.
When you create the IPS cells, presumably before that they come from mature cells, yeah?
Yeah, so they're coming from terminal skin cells or blood cells.
Those mature cells, will they have like epigenetic markers on them?
Yes.
So does that get removed as it goes through the IPS cell?
Yeah, so there's evidence that suggests that there's not lost. So
how does that affect when you look at diseases?
To be honest, it's not an area that I'm entirely familiar with, so I wouldn't be able to say too much about it, I'm afraid.
One thing that came up in one of the slides was the sort of risk of tumorigenicity,
Yeah.
so sort of potentially them becoming cancerous.
Yeah.
So could you explain a little bit more about how that happens and why, obviously, why that's a problem?
Yeah, so obviously there's a lot of excitement in taking someone's skin, for instance, who's known to have a genetic abnormality, using CRISPR-Cas9, for instance, to correct it, and then putting those stem cells back into the same person. The issue we've got there is that the stem cells' quality will be quite variable, and so putting those IPS cells back into a person can lead to abnormal behaviors. If they're not pure, for instance, or if they're not stable as a stem cell, they can go off and differentiate into other cell types. And that's why a lot of people are particularly cautious about speaking about this technology too loudly. I know in countries like China and Japan, they've actually already advanced to clinical trials, but at the back end of that, there's been a lot of work on confirming the quality of the stem cells that they've been using. So I think there was a talk by Shinya Yamanaka at a conference last week where they were talking about how they had to whole genome sequence the line, how they had to prove that it was stable in culture. They looked at various different donors. In the end, I think they've only got one clone that they know can be appropriate for clinical trials, but we're at very early stages with this. on the BBC Panorama.
There was a chap who had his sight restored by stem cells, is that right?
Yeah, so there was a BBC Panorama where I believe a guy, it may have been in Poland, where he had a slit in his spine, and they used IPS cell technology to they grafted and they actually regenerated the cut in his spine. And it was actually quite promising from what I heard from that story, because he was able to start feeling in his feet again and start to move his legs. But they didn't do a follow-up years down the line. So
we don't know what happened to him.
I don't know. We don't know.
So whether they're regulated, would we not have been allowed to do that in this country?
Not in this country, no.
Oh, that's what it is then. Okay.
Yeah.
Thank you so much. That was
Thank you very
really
much.
interesting.
Yeah. Thank you.
So our final session, we've only got about 15 minutes to do this, but I thought, well, we might as well just have a quick chat. So hopefully so far you've found something out that you didn't know before, and it's been really interesting. And Celine gave a marvellous overview of some of the applications of the stem cells, which is fantastic. But what I've wanted to do now is just finally really sort of showcase that there are some resources out there that could help support you in sort of what you've heard today, but also you can bring that to your students. So first of all, we've got your genome pages. So if you've got a pen on your desk, you'll see there's a URL on it. So feel free to take a pen. There are some in your bags as well. But we've just developed a brand new fact page on stem cells, which also includes what is an IPS cell and how that works. We do also have pages on CRISPR-Cas9 technology. So if there's anything that you sort of want to explain to your students, we've got some really nice diagrams that a bit like the ones that Tristan used, but that really break down the steps of CRISPR-Cas9 as well. So hopefully those 2 fact pages on your genome will be really good to support what you've heard today. But this is not the only place you can find information. Are you familiar with Eurostemcell? Some of you, yeah. So this website here, eurostemcell.org/education resources, is a really, really great site. It's got over 100 different resources, all on stem cells. So it's a really, really useful resource that I highly recommend. Some of the items on your table have come from that. So particularly this, which I find a good one. This is sort of almost like a comic about stem cells. So this is quite a good way to introduce stem cells to perhaps GCSE or even to A-level students, depends on what their learning styles, but this is quite a good one that's from there. There's also videos, animations and so on. To be fair, I would just have a quick look around it when you have some time, just to have a look at what it's got, because it is a real wealth of resources there. You can even sort of filter by it, filter by whether you want an activity, a debate or discussion, if you just want images, if you want a video, and so on. So there's a lot of resources on there. Other one that might be worth you having a look at is the STEM e-library as well. Are you familiar with this one? So it's basically type into Google STEM e-library, and this is produced by the National STEM Learning Centre. And if you literally just type in stem cells in the resources section, you will come up with over 600 different resources. You can filter by age range, and you can even filter by resource type as well. And they've got a whole collection. If you click on one of the collections like this, it has a whole section just on stem cells and some curated activities that they recommend for teaching as well. So again, this is quite a useful, well, not quite, a very useful site to use as well. And again, all of these are free. There's no charges associated with using these or accessing these resources. So definitely worth a look. So for the last sort of 15 minutes, I didn't know whether you wanted to sort of talk in your groups, and then we sort of feed back to the group, sort of any stem cell resources that you recommend that you've used in your teaching so far, or if you haven't, have a look at some of these ones that we put on the table, see what you think. Are they useful? Are they not useful? Would you use them? But also, is there any resources that you currently would really like, but just are not out there? And is that something that ourselves and Babram can help with? So perhaps if you want to talk amongst yourselves for, say, 5 to 8 minutes, and then we will feed back your ideas and we'll see what we can do. So I know you all love using a microphone, but whether anyone from the table wants to sort of just quickly feed back some ideas of what you've come up with, or any ideas? Go for it, Carol, go for it.
Well, got a wish list, which is, we were looking at this game, which has got a fabulous map about what mesoderm, endoderm and whatever do. And what would be lovely with it would be a very simplified set of protocols that what you have to do to an IPS to turn it into these different things, to give them, the kids, a practical idea of the fact there's almost like different recipes, and that it is actually...
And timeline.
And timeline,
yeah.
The things that we were learning up there.
Yeah.
Because it makes it real.
Yeah. Okay, so this, just sorry for the camera, so this is sort of come from what they call the Game of Life, the stem cell edition, and they've got a really nice diagram on here that shows, can't find it now, yeah, like you said, how all the cells differentiate between them. So yes, that would be really good to actually get a recipe book.
It
Yeah,
seems a bit magical and mysterious, and actually talking to a real scientist, you're learning there is a real practical thing that you do. You put it in a solution with various proteins or whatever, and
exactly.
it kind of helps them to link into the reality of it. Otherwise, I think it gets too mysterious.
Yeah, absolutely. And I think what was really interesting was the timelines, the time frames that Celine was talking about at the tour. The fact that, you know, it can take between 2 and 3 months to go from a skin cell to just, you know, the initial stem cell, and then another 3 months on top of that to then become, you know, the blood cell or the blastocyst.
Yes, because there's this idea you just put it in a Petri dish, do something to it, and suddenly, hey presto, you've got one, and that doesn't work like that.
Fantastic. So that's a really good suggestion. Anyone from another table want to contribute?
We didn't actually get as far as a wish list.
Oh, didn't you? Or just what you can, what you know?
Discussing the bits that we use.
Yeah, that'd be great. Can you sort of?
So I particularly like their I'm a Scientist, Get Me Out of Here, the stem cell debate cards. So we use that at GCSE. Really like those where they've got to take on the role of debating a particular individual. So the debate is, should the UK government fund embryonic stem cell research? Who are saying, you know, so you might have a sort of student, you might have their own, not sure about expressing their own views, they're taking on the role of the individuals that they get on the card. So bits where, you know, where they've got some information, then using it like that.
Fantastic.
One of the other things was I've used a lot of the resources on the yourgenome.org, and what I really like about them is they're ready to go. You don't have to tinker with them too much. They're just kind of there, which really saves time. There was one in particular looking at genotype and phenotype of zebrafish that I did with my sixth form, and they really got into it because they liked the fact it had been created by people who were actually doing the work. So it wasn't just sort of a textbook activity or something like that. So
okay,
yeah,
fantastic.
That was really good.
Brilliant. Anyone on this table got any favourite resources or any comments on what was on the...
I would say I particularly like this. I'm a big fan of comics.
Okay.
So yeah, I particularly like that as a resource, and I'll definitely be using that.
If on reflection on the way home goes, oh, I wish we could have a resource on that, please do email us. And anyone who's watching the video link, if there are resources that you are crying out for stem cells, please email us at engage@welcomeGenomeCampus.org, and we can pass that feedback on to Babram, and we will discuss whether it's possible to develop anything like this.